Good morning, dear students. Today we are moving to next chapter, that is nucleic acids. First, these nucleic acids are isolated by Frederick Mischer. It was from pus cells in 1869, and he named that substance as nuclein, as it is composed of an unknown phosphate substance. Till that time nobody know such type of phosphate, so they named that Frederick Mischer named it as nuclein, and then the term nucleic acid was introduced by Altman in 1899. Then purine and pyrimidine bases, two types of bases, that was discovered by Fischer in 1880. And next thing, histones we know that they are the basic proteins present in the chromatin for its organization, and these histones are recognized first by Kossel, a German scientist, and for that he got Nobel Prize, and for demonstrating the presence of two purines and two pyrimidines in nucleic acids. It is he who demonstrated that in the nucleic acids, two purines and two pyrimidines are existing. And who discovered two distinct nucleic acids, DNA and RNA, P.A. Levine. And for this nucleic acids, the DNA double helical model was proposed by James Watson and Francis H.C. Crick. These are some names of scientists dealt with significant findings in relation with nucleic acids. Then definition. What are nucleic acids? They are high molecular weight biopolymer. Biopolymer means it have some subunit. That basic unit is mononucleotide. Mononucleotide as repeated units, it form a big biopolymer. That biopolymer is nucleic acid. It is composed of carbon, hydrogen, oxygen, nitrogen and phosphorus. Types of nucleic acids. Two types are there. It's a very simple, from your lower classes itself you have learned that two types of nucleic acids are there, deoxyribonucleic acid and ribonucleic acid. Location. DNA, mostly the DNA present in the chromatin. Chromatin is the extended thread form of the chromosome. Extended form of the chromosome. In that chromatin exists what? DNA. Then what about RNA? RNA, 90% of the RNA is in cytoplasm as mRNA, tRNA and so many other forms. hnRNA are also there. So 90% of RNA are present in cytoplasm and 10% in nucleolus. Nucleolus seen inside the nucleus. Then along with that, extranuclear DNA also present. Extranuclear means the DNA other than that present in the nucleus. That is found in mitochondria and chloroplast. These are all different types of nucleic acids. And three components regarding its composition. Which are the three components? A phosphoric acid is there, a pentose sugar is there, and a nitrogenous base also there for nucleic acid. We are going to understand about each unit. First is phosphoric acid. Its molecular formula is H3PO4. It is given here. This phosphoric acid is a 3 monovalent, 1, 2, 3. 3 monovalent OH groups and 1 bivalent oxygen atom. Valent means valency. Valency means number of electrons, number of bonds they can form. That denotes its valency. That OH can form one bond So that's one monovalent. And phosphorus its valency is 3. That's why, sorry, its valency is 1 2 3 4 5. That's why 5 bonds are forming here. So that's about, since this OH are monovalent, so 3 monovalent OH groups we can find here, and also one bivalent. Bivalent means oxygen's valency is 2, and so 2 bonds are forming here. Nothing I need to bother much about the structure, only need to understand that phosphoric acid is composite structure. Regarding the structure, 3 monovalent HO groups and 1 bivalent O group is present. Then phosphoric acid, only that much is there. Then pentose sugar. Pentose means 5. Types of nucleic acids. Based on the type of pentose sugar it possesses, we can classify nucleic acids also 2 types. If the pentose sugar present is of D2-deoxyribose, then that nucleic acid is deoxyribose nucleic acid. If that pentose sugar is a ribose sugar, then that nucleic acid is known as ribonucleic acid. Then this D-ribose. D-ribose is actually the parent sugar. And from this, a derivative, its derivative is actually this deoxyribose. How deoxyribose is forming? By losing OH group in the second carbon position of ribose sugar, that I already explained to you in the previous chapter. Previous chapter means in the carbohydrate chapter. Then important property of the pentoses. They can form esters. They can form ester bond with phosphoric acid. That's an important property. And the bond that form, that's the bond that form the integral part of nucleic acid. And the main, that's skeleton of nucleic acid is formed with the help of a type of bond. That bond is phosphodiester bond. I will show you that bond here. This is one sugar, this is another sugar, this is another sugar. These are sugars, and these sugars are attached with the phosphoric acid. Also, so that here the 5th carbon is connecting with the phosphoric acid group, and here the 3rd carbon is connecting with the phosphoric. So these here one ester bond, here another ester bond. So 2 ester bond is there for connecting and one phosphoric acid group with the nucleic acid chain. So these 2 ester bond together we are calling it as phosphodiester bond. Okay. Then phosphoric acid group is over. Then pentose sugar is over. Then nitrogenous bases. Nitrogenous base types. Which are the types? Pyrimidine derivatives are there, and purine derivatives also there. Pyrimidine derivatives. And that pyrimidine derivatives actually they derived from parent heterocyclic compound called pyrimidine. Its structure is given here. This is the pyrimidine. To this pyrimidine structure, certain modifications are happening and variant forms are developing. Common pyrimidine derivatives are uracil, thymine, and cytosine. What about uracil? They are found only in RNA. They are white crystalline. Its molecular weight is 112.10 Daltons. Its melting point is 338 degree Celsius. Look here in the picture. This is a pyrimidine. What modification is happening to pyrimidine? In this position, one H is coming, and this position also one H is coming. And here one O group is added. That modification is happening to the basic pyrimidine structure. And that way what is forming uracil. Then another pyrimidine derivative is thymine. Yeah, that also can be found only in DNA. Its molecular weight is 126.13 Daltons. As it is first isolated from thymus. That's why it is named as thymine. Okay, it's a picture also given here. To this structure, what change is coming? A methyl group is added to this position. That's what the change. Mm, Next is cytosine. Cytosine is present in both RNA and DNA. It is also a white crystalline substance. Its molecular weight is 111.12 Daltons. Melting point is 320 to 325 degree Celsius. That about pyrimidine derivative. Then we are going to see purine derivative. Here the parent compound is purine. Purine structure is given here. It is a 6-membered pyrimidine ring fused to a 5-membered ring. This is the body structure. Then common purine derivatives are adenine and guanine. Adenine and guanine. Purine we can taking the first two letters, we will get pu. Then ag, ag in that first letter A for adenine, G for guanine. In that way we can memorize. If anybody feels difficult to memorize which are the purine forms, pu ag. In that way we can easily memorize the purine's name, adenine and guanine. Then adenine, this adenine is found in both RNA and DNA. Their white crystalline base, its molecular weight is 15.15 Dalton, and its melting point is 360 to 365 degree Celsius. Adenine, this is a structure. Here one amino group is adding. In that way adenine is forming. Then guanine. Guanine is also found in both RNA and DNA. It is also again colorless, insoluble crystalline substance. Molecular weight 51.15 Dalton. Melting point is between 360 to 365 degree Celsius. That much stable. And it is first isolated from bird guano, means bird's manure. So it is called so guanine. Some other purine derivatives also found. They are usually in plants. Caffeine. This caffeine is a purine derivative found in coffee and tea. Then theobromine is another derivative. Modified derivative is found in tea and coca. Then we are going to see some modified nitrogenous bases. First, we are going to see what modification are happening to pyrimidines, modified pyrimidines. Examples are 5-methyl cytosine. 5-methyl cytosine. To cytosine, fifth position, a methyl group is added. It is a common component of plant and animal DNA. And also another modified pyrimidine is appearing here in DNA of T, even bacteriophage. Bacteriophage means there are certain viruses that are infecting bacteria. So in such type of bacteriophage, their cytosine is not present. Instead, has a 5-hydroxymethyl cytosine and its glucoside derivatives in the place of normal cytosine. So, 5-hydroxyl methyl cytosine is a modified pyrimidine. Then pseudo-uracil. Pseudo-uracil means not actually the uracil, but what's the change here? The point of attachment to ribose is different. Uracils, its point of attachment to ribose is different. That difference make it pseudo-uracil. What's the difference? Here, the fifth carbon is joining with a ribose instead of the first nitrogen in the case of the normal uracil. That is the difference. Normal uracil, and first nitrogen is attaching with the ribose, but in pseudo-uracil, fifth carbon is attaching with the ribose group. Then modified purines. Modified purines, some are tRNA. In tRNA, some modified purines we can find there. And how that modified forms are forming through methylation by the addition of methyl group. These modified purines are obtaining. Example of such one modified purine is 6-methyl adenine. It is found in bacterial DNA. Then going to nucleosides. What are nucleosides? Nucleosides means it's a combination of a nitrogenous base. It can be purine or pyrimidine. It joined with a pentose sugar. How it combine with the sugar? Through a beta-glycosidic linkage. Beta-glycosidic linkage, everything we have learned in the carbohydrate chapter in very detail. Then beta-glycosidic linkage, that formation of that linkage, also some special points are there. In case of purines, this beta-glycosidic linkage is made between first carbon of sugar, either it may be ribose or deoxyribose, and the hydrogen atom of ninth nitrogen. So they are called the N9 glycosides. In the case of purines, that bond is forming between first carbon of sugar and hydrogen atom of ninth N. Then that nucleosides are called N9 glycosides. But in the case of pyrimidines, that bond is formed between first carbon and first nitrogen's hydrogen atom. So that case, nucleosides are called N1 glycosides. So compare the difference between them. Here the only difference is that that bond is forming between carbon 1. Both the case carbon 1 is involving in that bond formation. But in the case of the other part, there comes the difference. The hydrogen atom belonging to ninth nitrogen in the case of purines, and that of pyrimidines, the nitrogen atom of first position. Its hydrogen atom is involving in that glycosidic bond formation. The hydrolysis of nucleosides. Purine nucleosides can be very easily hydrolyzed by acid. But pyrimidine, they are much more stable. So prolonged treatment is necessary, and also that with strong acid. Then only we can hydrolyze pyrimidines. Then ribonucleosides and deoxyribonucleosides. Nucleosides containing ribose are called ribonucleosides, and nucleosides containing deoxyribose are called deoxyribonucleosides. That's the difference. Then unusual nucleoside is there. Unusual, when the nucleoside become unusual, when it have pseudo-uridine, and this pseudo-uridine is usually present in tRNA. What is the difference? Difference here also happens with respect to the beta-glycosidic linkage. And here that the linkage occurs between the first carbon of ribose and fifth carbon of uracil. Actually, that bond need to be formed between C1 of ribose and that of N3 of uracil. Instead of N3, fifth carbon is involving. That is the difference. Then nucleosides analogues as drugs. Some compounds that is similar to nucleosides that are having some modifications. They can be used as drugs. That analogues we are going to see now. Example for such analog is 3-acidodeoxythymidine. We can represent it as AZT. 2-3-dideoxycytidine can be shortened as DDC. Both are using in the treatment of AIDS. What is the mechanism? What mechanism involves as these nucleoside analogs are used as drug in the treatment for AIDS? We know that AIDS is caused by a virus called a human immunodeficiency virus. That virus, for its RNA replication, it depends on an enzyme called RNA dependent DNA polymerase. And that enzyme needs certain substrate to extend the newly forming DNA strand, newly forming RNA strand, sorry. Then that time in the place of actual substrate like this thymine triphosphate and also cytosine triphosphate. These are the actual substrate need to be incorporated to the growing RNA chain. But instead of its place, who is competing? A is a T. Azidodeoxythymidine is competing with the thymine triphosphate. And also in a similar way, this dideoxycytidine compete with the cytosine triphosphate. So in that way, it restricts the elongation of the RNA chain in the virus. In that way, that blocks its action in the patient. So it is used as a drug in HIV patients. Then we are moving to nucleotides. Nucleotides, what's the additional unit joining with the nucleoside? A phosphoric group. When a phosphoric group also add or join with the nucleoside, then it become nucleotide. So a nucleotide have three component parts: a pentose sugar, a base, and the phosphate group. If these three parts joins, then it become a nucleotide. Nucleoside means there is this phosphate group is absent, only sugar and base. Then positions of phosphorylation in ribose. In the case of ribose, in which position phosphate group is joining? Usually the positions are C2, C3 and C5. Why the C1 and C4 is not involving? Because it is very important points to form the Furanose ring. Furanose ring means only that ring-like structure involving 5 carbon. So in that ring formation, this C1 and C4 is very necessary. So they can't be used for phosphorylation. That's why in ribose, C2, C3 and C5. In these position, the phosphorylation can be possible. Then in the case of deoxyribose, in which position phosphorylation is happening? In the case of deoxyribose, C3 and C5 can involve in phosphorylation. C1 and C4 cannot be used for that purpose because it is a integral part for Furanose ring formation. And what about the C2? In the C2, it doesn't have a hydroxyl group, OH group. Only a H is there. So C2 also is not ideal to use for the phosphorylation. Then we are moving to functions of nucleotides. These nucleotides are acting as carriers of chemical energy. These nucleotides may have 1, 2 or 3 phosphate groups along with it. That means if you are taking the case of ATP, there is adenine triphosphate. Here adenine base is there and 3 phosphate group is there and 1 sugar is there. And similarly UTP also there. Uridine triphosphate. All these are storing huge amount of energy. So they are acting as carriers of chemical energy, and its hydrolysis breaking each bond between the breaking of each bond that hold a single phosphate group can liberate enormous amount of energy. So that is an exergonic process. So in that way we can tell that nucleotides are carriers of chemical energy. Then as components of enzyme factors, there are many cofactors and coenzymes. They have this nucleotides as its very important parts. Examples are coenzyme A, NAD plus and FAD. All these have adenosine as its one of the integral part. Without the adenosine, these cofactors and coenzymes cannot function properly. Similarly, nucleotides are acting as chemical messengers in cell signaling. Means if a cell need to respond corresponding to a hormone, it need the mediator. That mediator is called a second messenger, and those second messengers are all usually nucleotide. Example for such a second messenger is CAMP. Because whenever a cell got a trigger from by the way of a hormone, the response of that stimulus is conveying or happening through the formation of these type of second messengers. So that second messengers are all nucleotides. Okay, with this we finish today's class. Thank you.